The moment hydrogen peroxide touches potting soil, the surface can erupt with bubbles. It looks as if the soil has suddenly begun breathing. That reaction is real, but it does not automatically mean the roots are receiving a miracle dose of oxygen. Every disease is dying, or the plant has been rescued. To understand what is actually happening, follow one drop below the surface, because its most dramatic effect occurs in seconds, while the consequences can last much longer. Ordinary household peroxide is water with an extra oxygen atom in each molecule. That arrangement is unstable. When peroxide meets enzymes, metals, and organic material in the pot, it begins breaking apart into water and oxygen gas. The escaping gas forms the foam you see above the soil. More organic activity can create more fizz, but bubbles alone cannot identify fungus, prove root rot, or show that a treatment worked. They reveal a chemical reaction, not a diagnosis. The released oxygen does enter spaces around soil particles, and that matters because roots need oxygen to release energy from stored sugars. In a healthy pot, tiny pores contain a changing mixture of air and water. After watering, excess moisture drains and fresh air gradually returns. In a saturated pot, those pores remain filled with water. Roots consume the small amount of available oxygen, beneficial organisms compete for what remains, and the root zone becomes increasingly hostile. Peroxide can create a short pulse of oxygen inside that wet soil. Under special conditions, carefully controlled oxygen fertilization can help flooded or salinity-stressed plants, but the word short is crucial. Once the peroxide decomposes, the source is gone. It has not created a drainage hole, opened compacted mix, or made an oversized pot dry faster. If water is still trapped around the roots, the original oxygen shortage begins again. Now comes the part the fizz hides. Hydrogen peroxide is not just an oxygen carrier, it is an oxidizer. On contact, it can damage cellular components. That is why properly formulated peroxide products can suppress certain bacteria, fungi, and other microbial pests. It is also why concentration and exposure matter. The chemical does not read labels underground. It cannot neatly attack a harmful organism while promising to ignore every beneficial microbe and every tender root hair beside it. Fungus gnats show why this distinction matters. Their larvae live in moist organic media, where they feed mainly on fungi and decaying material and may also injure delicate roots. A mild peroxide drench can contact and kill some larvae. Cornell Cooperative Extension describes 1 part of ordinary 3% peroxide mixed with 4 parts water for this specific purpose. But that reaction does not catch the adults flying above the pot, and it does not remove the wet breeding conditions that attracted them. If the soil remains constantly damp, new eggs can restart the cycle after the bubbles disappear. Letting the upper layer dry when the plant tolerates it, trapping adults on yellow cards, and using a product containing BTI that is labeled for fungus-gnat larvae address different stages of the infestation. Peroxide can be a contact event. Moisture control is the environmental change that makes the pot less inviting. Root rot creates an even more convincing illusion. A plant wilts, the mix smells sour, and the roots turn brown or soft. Add peroxide and the pot foams, so it appears that oxygen is fighting the rot. It may reduce some organisms on surfaces it reaches, but it cannot turn dead root tissue alive again, and root rot is not one single disease with one universal cure. Waterlogged media, damaged roots, and several different pathogens can produce similar symptoms. The lasting repair is therefore physical as well as biological. Confirm that the symptoms really begin in the roots, remove tissue that is already dead when appropriate, use clean equipment, correct the watering pattern, and repot into a container and mix that drain properly for that species. If those conditions remain unchanged, repeatedly pouring oxidizer into the same wet pot treats the scene of the problem while rebuilding the problem underneath it. There is another popular claim: peroxide does not merely rescue plants, it makes healthy plants grow faster. A USDA study tested several very dilute peroxide solutions on potted nasturtiums. The lowest treatment did not produce a significant overall growth advantage over ordinary water, while 2 higher treatments reduced foliage, dry weight, and flower numbers. That was one plant species under one set of conditions, so it does not answer every horticultural question. but it exposes the dangerous assumption that more bubbles or more peroxide must mean more growth. The soil is also not an empty brown sponge. It is a living network of bacteria, fungi, decomposers, and, in some mixes, organisms that exchange nutrients with roots. Oxidation is useful precisely because it can alter living cells. Research in diseased replant soil has found that peroxide treatment can suppress some fusarium while also changing the microbial community. That may be useful in a controlled agricultural treatment, but sterilizing broadly is not automatically an improvement. Removing competitors and partners can change what grows back after the peroxide is gone. This is why a professional horticultural peroxide product and a brown pharmacy bottle should not be treated as interchangeable. Registered crop products specify a concentration, target, application method, protective equipment, and crop use. Some combine peroxide with other active ingredients. A recipe copied from a different product, a hydroponic reservoir, or a commercial greenhouse may be inappropriate for a houseplant growing in organic potting mix. Even 3% deserves respect. It describes the concentration inside the bottle, not the concentration a particular root should receive. Never substitute concentrated food grade or industrial peroxide into a household recipe. Concentrated solutions can cause severe burns. Keep peroxide in its original light-blocking container, protect skin and eyes according to the label, and never mix it with bleach, vinegar, or another cleaner. A plant treatment is still a chemical exposure. Before anything enters the soil, diagnose the problem. Tiny flies suggest gnats. A pot that stays heavy suggests trapped water. Soft, dark roots demand inspection. For a healthy plant with no identified problem, routine peroxide watering offers little reason to accept the risk. For fungus gnats, a carefully diluted, limited treatment may knock down contacted larvae, but it should sit inside a larger plan. For a failing root system, correcting drainage and removing the cause matters more than creating foam. And when a pesticide claim is being made, the product label, not a viral recipe, determines where and how that product can legally and safely be used. So, what does hydrogen peroxide actually do inside plant soil? First, it reacts. It breaks down rapidly, releases oxygen gas, and creates the bubbles that make the treatment look powerful. At the same time, it oxidizes living material it contacts, which may include pests and pathogens, but may also include beneficial organisms and sensitive roots. Then the reaction fades, leaving behind the same pot, the same soil structure, and the same watering habits unless those were changed separately. The bubbles are therefore neither proof of a cure nor proof of damage. They are a brief chemical event. The real outcome depends on the concentration, the plant, the target, the organisms in the soil, and, above all, the condition that existed before the bottle was opened. Peroxide can sometimes be a tool. It is not new drainage, permanent aeration, or a substitute for diagnosis. What saves the root zone is not the loudest reaction above the soil, but the environment that remains after the fizz is gone.